Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be talking about the last part of the topic of toxicology in the crime laboratory. So I think in the last session I had ended with the role of alcohol inside our body. Role means the action, the physiological action which obviously leads to pathological effects and adverse effects inside the body, particularly with respect to the respiratory system and the circulatory system. Okay. So in this session, since this is the last session of this topic of toxicology, so here I would mainly like to focus on the alcohol levels in the body and what are the different techniques by which we can actually detect the level of alcohol in the body. Okay, the different tests or the different techniques that are done. So specifically the breather tests and the breath analyzer tests, okay, and also the relation of alcohol with law. So firstly beginning with alcohol levels, so the alcohol concentration slowly decreases, means obviously after consuming it there will be a spike, then there will be a decrease, then again there will be a type of like a slight spike in the graph, and finally it will be decreasing and zero level will be attained. Okay, so that is the final scenario. So basically it's a graph which is having some shifts. So there are some factors which will be determining the rate at which alcohol will be absorbed inside our body. What are the factors? Number one, time taken to consume the drink. More time taken, more will be the rate of alcohol that is absorbed. Obviously, because the time is more. So the body is getting more time to absorb that alcohol, isn't it? Then the alcohol content. That is obviously directly proportional. More the alcohol content, more will be the rate at which absorption will occur. Then the amount consumed and the food, obviously amount consumed is again something which will be directly proportional, and the food that is present in the stomach, that will also be determining the rate at which alcohol is absorbed. I will be showing you a graph which will help you to understand in more details vividly about how basically this food factor is going to affect, because more food is there in the stomach, less absorption will be there, less amount of alcohol concentration will reflect in the blood, okay, irrespective of what amount that person is taking. But if the stomach is empty when that person is drinking, then the level of alcohol in the blood will shoot. It will be rising at a faster rate. Okay. Okay. So moving forward, so elimination of alcohol throughout the body will be mainly accomplished by the liver, as I mentioned in the previous session also. 92 to 98%, sorry, 97 to 98% will be detoxified or eliminated by the liver with the help of the enzyme alcohol dehydrogenase by detoxification mainly, sorry, by oxidation mainly, and the remaining 2 to 5% will be eliminated directly in the form of alcohol only, not after being detoxified. It will be eliminated in the form of alcohol only directly by through the sweat, that is perspiration, through the urine, and also through our breathing or respiration process. Okay, so that is exactly what they are saying over here. Elimination of alcohol throughout the body is accomplished through oxidation and excretion. Oxidation takes place mostly entirely in the liver, while alcohol is excreted unchanged in the breath, urine, and perspiration. Exactly what I mentioned. The extent to which an individual may be under the influence of alcohol is usually determined by either measuring the quantity of alcohol present in the blood system or by measuring the alcohol content in the breath. So in order to, as forensic investigators, in order to to study, or in order to get an idea about the changes in the behavior of the suspected criminal or maybe the victim, because in many cases it can happen that the victim was forced to have alcohol by the criminal or by the suspect, isn't it? So in order to judge, in order to understand the change in the behavior of that person, we have two options. Either we can measure the amount of alcohol that is present in the blood of that person, or we can measure the alcohol content present in the breath of that person. Okay, so now see, it's not always possible. Say for example, the criminal or the victim has been caught in the middle of the road, so it's not possible to go and collect blood in the middle of the road, from a person, isn't it? The proper environment is not there, the proper personnel, the proper equipments is not there, obviously, in the middle of the road. So we need to take uh, preparation and prepare an entire procedure or a protocol, etc., etc. So that is always not possible. But to detect the alcohol content in the breath of that person, that can be possible even inside the middle of the road, if we have a proper equipment or an instrument or a machine. And that is exactly where the importance of breather tests or breath analyzer tests comes, okay, which I will be talking about in the upcoming slides. So let's move forward. So this is basically a graph that I have given so that you can understand the relationship between the alcohol level and whether the alcohol is taken in an empty stomach or a full stomach. Okay, so it makes a huge difference basically. So first let's see what they're saying over here. Experimental evidence has verified that the amount of alcohol exhaled in the breath is directly proportional to the blood concentration. Means whatever alcohol amount or alcohol content is detected in the breath of that person, that means that equal amount of alcohol is actually present at that point of time in the blood of that person. Okay, so this graph or this image that I have given over here, it basically helps us to understand or to get an idea about the blood alcohol level after drinking. Okay, so see, first let's focus on the left hand side and then let's focus on the right hand side. So on the left hand side they are showing the scenario of a person who has taken alcohol in an empty stomach, while in the right hand side they are focusing on the person who has taken alcohol on a full stomach. So see, what's the basic difference? So they are showing three main radars or scales over here. So one is the body weight, second is the ounces of liquor that is consumed. Liquor means alcohol that is consumed. And the third scale is showing the maximum blood alcohol concentration in that person's blood or in that person's body. In the right hand side also the radars are the same, but the scenario is different. So first let's see on the left hand side what is happening. Say for example we are considering a person, male or female irrespective of the gender. The weight of that person is around 150 lb, means 72 to 75 kgs weight. So if that person is consuming alcohol on an empty stomach, say for example around 8 ounces of alcohol, the blood alcohol concentration is coming to be around 0.15. But that same person means person with the same weight, when he is having alcohol in a full stomach, in fact more amount of alcohol, here it's 10 and here it's 10, sorry, here it's 8 and here it's 10, okay, 10 ounces of alcohol, then that same weight person, that person weighing the same weight, will be having the same alcohol concentration only. Means what? Even if you are taking a more amount, even if a person is consuming more amount of alcohol in a full stomach, the level of alcohol concentration is the same if the same person had consumed more amount of alcohol, sorry, less amount of alcohol, but in an empty stomach. So what is the moral of the story? That the empty stomach is actually aggravating the effect of alcohol in the body. It's actually aggravating the absorption and also increasing the amount of alcohol that is there in the blood. Means it's rapidly getting absorbed because the stomach is empty. Whatever thing the stomach is getting is alcohol only. So the stomach will be rapidly starting to absorb the alcohol because the stomach doesn't know that whether the alcohol is harmful or not for the body. Okay, the stomach is thinking that okay, I have gotten food, now I have to metabolize, now I have to absorb or digest this food. So obviously the absorption will be rapid and hence it will be reflected in the blood concentration in a much higher rate. Okay, so let's see the next scenario also. So in that case, the same person weighing around 74 to 75 kgs. Now this person is taking, say for example, much less amount, 5 ounces of alcohol, but the alcohol concentration in his blood is coming to be 0.10. But again, when this same person is taking more ounce of alcohol, that is around 7, here it's 5, here it's 7. But see, the alcohol concentration is same, 0.10. Alcohol concentration in blood is same. Why? Because now this person is taking it on a full stomach instead of an empty stomach. Okay, same in the third scenario also. 3 ounces on empty stomach, alcohol concentration 0.05. But around 5 ounces on full stomach, alcohol concentration still the same, 0.051. Okay, so that is the moral of the story. Now coming to the main part or the main content of today's topic, that is breath testers. So breath testers are what, as you can exactly see in the image. So it looks like somewhat like a walkie-talkie, isn't it? So there's a pipe that is connected. So the police or the investigating officer that is there, he or she will just hold it near your mouth. You need to blow to it, and it will be detecting your breath instantly and giving you the result on the display board, whether it's positive or negative. So how it's basically working, we will be talking about that in detail. So many types of breath testers are designed to analyze a set volume of breath. The captured breath will be exposed to infrared light, and from there you will be getting the result that is reflected, the numerical value. So basically what is the principle of this breath testers? Is the degree of the interaction of the light with alcohol in the captured breath sample that allows the instrument to measure a blood alcohol concentration in breath. So basically it's converting the alcohol content to a light signal. Okay, so the degree of absorption of light will be directly proportional to the alcohol concentration. Basically that is the funda. So some breath testing devices also use fuel cells. Okay, so these are actually the breath test instruments which were found in the earlier, zamanas, earlier days, when actually this breath analyzers came into display or came into role. So see, this is exactly what it looked like in the 1990s. A device, so till the 1990s, it was not like until 1990, means before 1990 it was not in rapid use. After 1990 or from 1990 it actually came into active use. So it is a device for collecting and measuring the alcohol content of alveolar breath, means breath which is directly coming from the lungs. Okay, so now talking about the breath analyzer test. So what is basically the principle or the procedure? We will be talking about that. So basically the subject, means the person who is convicted or who is suspected to have consumed alcohol, blows into a mouthpiece that leads to a metal cylinder which causes a piston to rise to a height that exposes two vent holes. When collection of alveolar breath is complete, the piston drops back into place, trapping the collected of about around 52.5 ml or 1/40th of 2100 ml of alveolar breath. Because we all know, again this is something I've talked about in the earlier session, that the lung capacity is around 2 liters. So 2 liters ka 1/40th of 2 liters, which is around 52.5 ml. That will be the capacity of the uh, tube that is there, or the metal cylinder that is there. Okay. So this trapped alveolar breath can then be analyzed as it passes into a glass ampule containing 3 ml of 0.025% potassium dichromate and 0.025% of silver nitrate in sulfuric acid and water. So see, whatever light sensing or light absorption is there, there has to be some kind or some type of chemical reagent there which will react with the alcohol so that the light can be absorbed. Okay, and what chemical reagent is that? That is potassium dichromate and sulfuric acid along with water. So I will just highlight this. See, percentage, if even if you cannot remember, it's okay, but what are the components that are there in the glass ampule which are actually reacting with the alcohol? That thing you have to remember, and also the amount, that is 3 ml. So inside the glass ampule there will be 3 ml of potassium dichromate as well as silver nitrate, which will be present in suspension in a solution of sulfuric acid and water. So any alcohol which will be present in the sample, if any, if at all, it doesn't necessarily mean that if the person is suspected, it's sure that that person has consumed alcohol, isn't it? It can be negative also. So if at all any alcohol is present in the alveolar breath sample, then it will be dissolving itself in the dichromate solution and it will be oxidized to acetic acid. So that is basically the chemical reaction that will occur. I will be showing you the chemical reaction so that it gets clearer to you. So after this oxidation process, what will happen? It will be destroying the potassium dichromate, the extent of which will be measured by the breath analyzer, and it relates to the quantity of alcohol passed into the ampule. Means the extent to which this potassium dichromate will be destroyed by the formation of acetic acid will actually be directly proportional to the amount of alcohol which was present in the original alveolar breath sample. So this is a spectrophotometer specifically designed to measure the absorption of light passing through the potassium dichromate solution at a single wavelength. Okay, so here comes the role of the light. It will be passing through the potassium dichromate solution and generating the signal, which will be detected by the spectrophotometer in the form of a numerical value. Okay, so if at all the potassium dichromate is formed, then only we will be getting the result. Okay, so see, this is actually what is happening. So we have potassium dichromate and we have sulfuric acid solution, and say for example if the result is positive, then that means in the alveolar breath alcohol is present, which is nothing but ethyl alcohol, C2H5OH. So these three things will be reacting and producing chromium sulfate, potassium sulfate, acetic acid and water. So this acetic acid will be going and destroying or destructing the potassium dichromate, which will be reflected by light absorption. So that is the basic fund. Okay. So this potassium dichromate that is there, it is yellow in color and it will be absorbing visible light wavelength of 420 nanometer. So the spectrophotometer has to be set between the wavelength of 420 nanometer, in the range of the wavelength of 420 nanometer, and it will be, then only it will be able to absorb the yellow color which will be produced if the test is positive. So the breath analyzer will indirectly determine the quantity of alcohol which is consumed by measuring the absorption of light by potassium dichromate before and after its reaction to alcohol. So before its reaction with alcohol, it will be intact, it will not be destroyed, isn't it? But after its reaction with alcohol, it will be converted to potassium sulfide. So that will be giving you the result, and then you will just have to compare. Okay, that means you don't have to compare, the machine itself will compare and give you the result. So if there is at all any kind of difference, then that means it's positive, and if there is no amount of difference in the light absorption wavelength or in the reading of the spectrometer, then that means there is no alcohol present. Okay. Nextly coming, so these breath analyzer tests are very useful, they are very handy, they can be performed at any place at any time of the day. Okay, and also the person who has to perform the test, like who has to take part in performing the test, also will not be uh, like it will not be hazardous neither to the person who is performing the test, or neither to the person who is assigned the duty of performing the test. Okay, so it's a very suitable and friendly type of a test. Other tests that can also be done for detecting alcohol content will be the field sobriety tests. Okay, so among the field sobriety tests, various different tests are there. So number one will be the psychophysical tests, number two will be the walk and turn test, and number three will be the one leg stand test. So let's see what are these tests all about. Okay, so talking about the psychophysical tests, so they will be very much reliable and effective. So what we will do is we will be doing the nystagmus test. Okay, so nystagmus basically means rapid movements of eyes. Okay, which are particularly seen in drunk persons. Okay, so it's basically known as horizontal gaze nystagmus, means whether the person can look in the horizontal direction accordingly like this. Okay, see the finger is moved like this and it's seen whether the person can actually coordinate that movement with the eyes. So involuntary jerking of eye as it moves to side, as intoxication increases, the ability of the eye to follow an object as it moves to the side without jerking decreases. If BAC means the total maximum alcohol content in the blood is at 0.1%, then eye jerking will begin at 45-degree angle. As BAC increases jerking, it will begin at increasingly small angles. Okay, so basically it's a measurement that if BAC is this much, then jerking will begin at this much angle. So that is about the nystagmus test. The next test that we can do, sorry, is the walk and turn test. So it's basically a divided attention task, means whether the person is actually able to give his undivided attention or not to a particular task. So because alcohol, as I mentioned when I talked about the different physiological or pathological effects of alcohol on the body, mainly the brain, isn't it? I showed you a nice figure at the end of the last session of the brain, of the different parts of the brain, and what are the important functions of the different parts of the brain. And since the effect of alcohol is mainly on the brain of the body, so it will be affecting all the different functions of the brain, all the different functional zones of the brain, okay, functional centers of the brain. So in this case, in the walk and turn test, the subject's ability to comprehend and simultaneously carry out more than two instructions is basically tested. So this ability decreases with increasing consumption of alcohol, because obviously your attention will be decreased. So it will not be able to respond properly to various instructions that are given to you, and specifically if more than one instruction is given at the same time simultaneously. And one leg stand, the last test, that is also a very tough test for a drunk person, isn't it? Because again, it's a divided attention task. You have to, because uh, one leg stand will mainly, identify your effect on the cerebellum, because cerebellum is the center of the brain, or the part of the brain which deals with the coordination, the balance, etc. So that will be disturbed obviously if the cerebellum is hit by alcohol. Okay. Okay, so these are the different images that are showing how these different tests can be done. So this is, I think, the walk and turn one. This is the nystagmus one. Okay, and this is again the one leg stand one. Okay, this is again nystagmus one. At an angle of 45 degrees, it's seeing that whether the movement is rapid or not. Okay, and then other tests that we can do is gas chromatography testing. So this is again a diagram that I have shown over here. So after gas chromatograph, this is the particular chromatogram that we will get. Okay, typical like a graph or a curve. And then we need to do mass spectrometer in order to confirm or finalize the results. Okay, confirmatory test basically. So after this mass spectrometry, we will be getting the different spectra from these different chromatograms. So the see, the chromatograms are having the different peaks A, B, C, D, and from these peaks, so according to these different peaks, we will be getting the different spectra. So gas chromatography will be offering the toxicologists the most widely used approach for determining alcohol levels in the blood. Now coming to the last section of this topic, that is alcohol and law. So the American Medical Association and the National Safety Council have been able to exert considerable influence in convincing the states to establish uniform and reasonable blood alcohol standards. So basically the rules should be uniform in all the parts of the country, or I would say all the parts of the world. Anyways, between 1939 and 1964, a person having a blood alcohol level in excess of 0.15% of weight per volume was to be considered under the influence, which was lowered to be 0.1% by 1965. In 1972, the impairment level was recommended to be lowered again to 0.08% weight per volume. It remains there today. So, pehle it was 0.15%. Now it has been amended to 0.08%, means which is much lower. Okay. To prevent a person's refusal to take a test for alcohol consumption, the National Highway Traffic Safety Administration recommended an implied consent law. Adopted by all states by 1973, this law states that the operation of a motor vehicle on a public highway automatically carries with it the stipulation that a driver will submit for a test for alcohol intoxication if required, or be subject to loss of the license. So this law is actually very good. I don't know how much it is followed or not, because drink driving is something which must be prohibited at any cost. Okay, and nowadays there are very various strict rules and fines that the government have come up with. And again, this is something I always say, that the government cannot take the entire responsibility if we are not taking our own responsibility, if we are not concerned about our own safety, particularly when we are traveling, when we are driving on the roads. So then who will take the responsibility, isn't it? We should be utmost responsible for our own safety. So the government can at the most make some rules and regulations, some laws, some they can impose some fine, but that's it. The ultimate safety is something that the person has to think of. Okay, if he loves himself or herself and his or her family. Okay, so drink driving is something which can be very fatal, which actually is very fatal, because driving is a very good exercise. It's an exercise where all the different centers of the brain are active. Your eyes are active, your senses are active, your ears are active, okay, your legs are active, the entire brain is very much active. So, and again, alcohol is a toxicant which is actually depressing the brain. So it's depressing the center which is actually required to be active for driving. So it's, you know, it's, they are like what to say, perfect enemies to each other, alcohol and driving. So whenever a person is drinking and driving, that means he or she is taking his or her life for granted, and that should never be the case. Okay, so with that I would just like to end this session. Please like, share and subscribe ITLS Academy. We will soon be coming up with the next session. Thank you and goodbye.